The idea of antibiotic resistance, it's something that's really important and really scary. And even though you hear about it in the news, I don't know that it gets the press that it should and exactly how scary it is. So it seems like a great place to put my research interests and actually make a contribution. There's a couple problems that feed into the issue of antibiotic resistance. So one is that the antibiotics that we have are currently being maybe overused. At the same time, the development of new antibiotics hasn't kept pace, so we don't have new antibiotics to replace the ones that bacteria are becoming resistant to. And so if you have a single bacteria in your body that is resistant to a specific antibiotic, the antibiotic will kill all the other bacteria but leave that single one. That one that is resistant is now able to multiply to create more and more daughter cells that are also resistant to that antibiotic. So we do several different things within our research. So we start by actually growing bacteria, purifying either proteins or outer membrane vesicles. We also grow human cells, and we sort of combine the two and see what happens when we look at what these toxins are doing to the human cells or what the outer membrane vesicles are doing to human cells. We also synthesize molecules called peptides, which we can use to prevent the recognition of certain molecules. And so we use these peptides to show that we can inhibit the recognition of the toxin or the vesicle with the human cell. Our goal is to understand those mechanisms so that we can block those specific steps to prevent disease. So specifically, we've been working with outer membrane vesicles recently, which bacteria use to deliver specific molecules to the host to communicate with the human body. So these vesicles contain specific molecules called virulence factors, and these virulence factors are molecules that allow the bacteria to cause disease. So there are some ideas in the field that an antivirulence approach has a really significant advantage over antibiotics in that the bacteria will become resistant at a much slower rate than they do to current antibiotics. The idea is that if we can stop those virulence factors from doing what they do, the bacteria are going to become more uncomfortable, less able to divide and grow within the body. And this gives the immune system an opportunity to clear the infection. We're sort of boosting the immune system's ability to clear the infection. The NSF Career Award is an award that's given to assistant professors, and the idea is that it's supposed to help you get your career started. And so they provide funding for your research. You also have to demonstrate that you're a talented educator, and you also have to show broader impacts of both your research and your teaching. So I've participated in a couple programs that we have here, including the Choices and the Options program. And what we've done there is create and run some demonstrations about chemical engineering and what exactly chemical engineers do, so that these students are just aware of that career option. So the funding will be used primarily for the research in my lab, so it's really going to help us get started in a different path within my lab, which I'm really excited about.